Hello, class. Very good evening, very warm welcome to everyone. Sorry I got a bit late. It's just was into something. And let's start the class. Oops, speakers, headphones got out. So today is 28th. No, everything is fine. So let's people join in, and then we start our next session. So I hope everyone has joined now by now. So, student, there is a some good news or bad news. It's up to you how you wanna seek it. So somehow I have to leave from here on 1st of November from my hometown so for some my work. So we have today 29, 30 and 31st. And I don't know whether I can get the internet access at that place. So I would like to finish things with you maybe by 31st of October. So please cooperate with me. I will be really appreciate that because we started on 25th. Today is the fourth day. So fourth, fifth, sixth, seventh. So we have 31st of October to finish with our sessions. And so let's try. I will try to bring everything to be compensate things in between these days. If not, if I'm not able to take class from 1st November onwards, I will see if I'm able to get Wi-Fi at that place. So some days have to be compensate. So let's see. But your whole course of my MBBT, your all workshops, all videos, all concerns is my responsibility, and I will take care of that. That whenever you are leaving, you leave with a satisfaction, and all the things that we are learning, you learn it properly, not in fast way. So okay, so we were doing yesterday with lot of molecular biology techniques. Yesterday we were finished with of PCR, gel electrophoresis, blotting techniques, real-time PCRs, microarrays, recombinant DNA technologies. And today onwards, we have to start with Sanger sequencing and next generation sequencing. So we will be dealing with that part and method to study gene functions. So somebody is asking questions. Would we still allow to submit our reviews by 9th, or no, you can take that time. It's just I might be not available of the internet. Emergency I have to leave some place, and there I might or might not able to get internet. So I don't want to make sure that when I leave from here, you don't I'm not. So in that case, I will try to compensate to do things done earlier with some extra classes, and then everything will be recorded with you so that you can have things to be done on proper way. Okay, so maybe we can class of genetics before this MBBT. I will see how we will manage, but at the moment we will continue with these classes as per it is, not so things upside down. So this all we did. So now comes the next part, sequencing, that is Sanger sequencing. So here you can see you have, and then to do so, you make a duplicate like sequence of that. Then you add DNA polymerase, DATPs, GGPs, GCTPs, DDTPs, and GTPs in the low concentration. This is guanine triphosphate, cytosine, tyrosine triphosphate, adenosine triphosphates, and DD GTPs, they are in the low concentrations. So you have all these things. So you have deoxyribonucleic nucleoside triphosphate, DNTPs, and DDTNTPs. So I will show you this small video about how that DNA Sanger sequencing method is done. So let's go through this. So I hope this was clear. DNA Sanger sequencing with the DDNPs, how things were happening. So next generation sequencing, was the old one, but mostly Sanger is not being used. Most likely we are using this technique of next generation sequencing. Um, So something like Roche, Illumina, Applied Biosystems, Helicos, Specific Biosciences, Life Technologies, Iron Torrent. So they are the various things are available, and these are the various features of these next generation sequencing. Roche, Illumina, Live, Ploninator, Helicos, Specific Biosciences. And next generation sequencing looks like so mainly three are there, that is Roche, Illumina and Solid. The chemistry behind the Roche is pyrosequencing. For the Illumina it's polymerase based, and Solid is ligation based. So, and the paired base ends are 3 kilo base pairs, 200 base pairs and 3 kilo base pairs, with mega base pair run to be 100 MB, 1300 MB, 3000 MB. Time run is 7 hours, 4 days, 5 days, with read length has 250 base pairs, 32 to 40 base pairs, 35 base pairs. And cost per run is 3439, 38950, 17447. So this is extremely expensive. But cost per MB is 84.39, 5.97, and here is 5.81. So Roche 454 workflow looks like this with the pyrosequencing reactions, where you have polymerase is being attached, annealed, and then APS and PPIs are added, then sulfurase, luciferase are added, the ATP luciferin reaction is done, then you have light luciferin out of it. And this is the Illumina Solexa workflow, where the ligators, adapters actually are working, and they're making the reactions to be illuminate like that. So we will go about them in each of these details in this respective video. So let's go. So it will explain each video very nice and simple way.
Next-generation sequencing, or NGS, is a powerful platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. This powerful tool is revolutionizing fields such as personalized medicine, genetic diseases, and clinical diagnostics by offering a high-throughput option with the capability to sequence multiple individuals at the same time. Sanger sequencing, first developed in the 1900s, is a gold standard for DNA sequencing, and it is still used today extensively for routine sequencing applications and to validate NGS data. It utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from its 3' end. Deoxynucleotides, or simply nucleotides, are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 dideoxynucleotides, one for each DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1. They lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2, each dideoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the dideoxynucleotides. The reaction mixtures are loaded on the sequencing machine, either manually onto slab gels or automatically with capillaries, and are electrophoresed to separate the DNA molecules by size. The DNA sequence is read through the fluorescent emission of the dideoxynucleotide as it flows through the gel. Modern-day Sanger sequencing instruments use capillary-based automated electrophoresis, which typically analyzes 8 to 96 sequencing reactions simultaneously. Next-generation sequencing systems have been introduced in the past decade that allow for massively parallel sequencing reactions. These systems are capable of analyzing millions or even billions of sequencing reactions at the same time. Although different machines have been developed with various differing technical details, they all share some common features. 1. Sample preparation. All next-generation sequencing platforms require a library obtained either by amplification or ligation with custom adapter sequences. 2. Sequencing machines. Each library fragment is amplified on a solid surface with covalently attached DNA linkers that hybridize the library adapters. This amplification creates clusters of DNA, each originating from a single library fragment. Each cluster will act as an individual sequencing reaction. And 3. Data output. Each machine provides the raw data at the end of the sequencing run. This raw data is a collection of DNA sequences that were generated at each cluster. The differences between the different next-generation sequencing platforms lie mainly in the technical details of the sequencing reaction and can be categorized in 4 groups: pyrosequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor sequencing. In pyrosequencing, the sequencing reaction is monitored through the release of a pyrophosphate during each nucleotide incorporation. The released pyrophosphate is used in a series of chemical reactions resulting in the generation of light. Light emission is detected by a camera which records the appropriate sequences of the cluster. The sequencing proceeds by incubating one base at a time, measuring the light emission, if any, degrading the unincorporated bases, and then the addition of another base. This technology is capable of generating large read lengths, much comparable to the read length of Sanger sequencing. However, high reagent cost and high error rate over strings of 6 or more homopolymers have reduced its applications. For more details on the technical aspect of this technology, please visit our knowledge base at the link provided in the description below. Sequencing by synthesis utilizes the step-by-step incorporation of reversibly fluorescent and terminated nucleotides for DNA sequencing and is used by the Illumina NGS platforms. All 4 nucleotides are added to the sequencing chip at the same time, and after nucleotide incorporation, the remaining DNA bases are washed away. The fluorescent signal is read at each cluster and recorded. Both the fluorescent molecule and the terminator group are then cleaved and washed away. This process is repeated until the sequencing reaction is complete. This system is able to overcome the disadvantages of the pyrosequencing system by only incorporating a single nucleotide at a time. However, as the sequencing reaction proceeds, the error rate of the machine also increases. This is due to incomplete removal of the fluorescent signal, which leads to higher background noise levels. Our NGS, an introduction knowledge base, provides more technical details about this technology. Sequencing by ligation is different from the other 2 methods since it does not utilize a DNA polymerase to incorporate nucleotides. Instead, it relies on 16 octamer oligonucleotide probes, each with one of 4 fluorescent dyes attached to its 5' end that are ligated to one another. Each octamer consists of 2 probe-specific bases and 6 degenerate bases. The sequencing reaction commences by binding of the primer to the adapter sequence and then hybridization of the appropriate probe. This hybridization of the probe is guided by the 2 probe-specific bases and upon annealing is ligated to the primer sequence through a DNA ligase. Unbound oligonucleotides are washed away, then the signal is detected and recorded. After that, the fluorescent signal, along with the last 3 bases of the octamer probe, are cleaved, and then the next cycle commences. After approximately 7 cycles of ligation, the DNA strand is denatured and another sequencing primer, offset by 1 base from the previous primer, is used to repeat these steps. In total, 5 sequencing primers are used. The major disadvantage of this technology is the very short sequencing reads generated. Ion semiconductor sequencing utilizes the release of hydrogen ions during the sequencing reaction to detect the sequence of a cluster. Each cluster is located directly above a semiconductor transistor, which is capable of detecting changes in the pH of the solution. During nucleotide incorporation, a single hydrogen ion is released into the solution and it is detected by the semiconductor. The sequencing reaction itself proceeds similarly to pyrosequencing, but at a fraction of the cost. Please view our knowledge base for further details on ion semiconductor sequencing and the sequencing by ligation techniques. In order to be able to showcase and compare the different technical aspects of each of the above technologies, the number of coverage that each run generates when sequencing the whole human, mouse, Arabidopsis thaliana, and E. coli genomes are calculated and presented here. The presented data is based on the most powerful machines of each technology. Further details can be found on our knowledge base. For whole genome sequencing data to be useful, a minimum of 30 times coverage is required. As it can be seen, the pyrosequencing method is only able to sequence the E. coli genome at enough coverage to result in valid data. The sequencing by synthesis method, which is the most popular method currently on the market, is able to generate hundreds of coverage per run. In fact, with this machine, it is possible to sequence 15 individuals within 3.5 days. The sequencing by ligation method also generates enough coverage for all genomes to be used. However, it isn't capable of generating nearly as much output as the Illumina HiSeq machines. The iron proton machine is used mostly in clinical settings because it is able to provide a sufficient size output within 2 hours. ABM offers a wide range of next-generation sequencing services. These include whole genome sequencing, exome sequencing, RNA sequencing, disease panels, lane rentals, and much more. To be able to access our services, please visit our website at www.abmgood.com, and from there, click on the NGS Sequencing Services link. This will load our NGS service webpage, which details all of our available services. Clicking on a service of interest will showcase the technical details, pricing, and bioinformatics solutions that are related to that particular service. Please leave your questions and comments below, and we will answer them as soon as possible. For more information, please visit our knowledge base at the link provided below. Thank you for watching.
So now we are done with the all the sequencing. So there are some applications that you can go for with the genomes, resequencing human exons, small and long RNA profiling, chip sequencing, DNA methylation, polysomal RNA, origin of replications, whole genome associations, all these other various applications of it. And further, to study the function of a gene protein, some basic principles you have to take care. That is, you have a mutant and you have a wild type. So you have genetic analysis to produce a clone gene of DNA sequencing, and then you have a protein produced in the localization of biochemical studies, determination of the structure. So you have gene inactivation and database search to identify protein coding sequencing and PCR isolation of corresponding genes. So there are some terms that you have to remember. That is in vivo, that is in living organisms, ex vivo, in an organ explants, in vitro, in reagent tube, and in silico is the computerized based simulation. So there are advantages of working with culture cells over intact organisms. So more homogeneous than cells in controlled experimental conditions, can isolate single cells to grow in colony of genetically homogeneous clones, and ethically non-controversials. They are non-controversials also. So examples are E. coli and yeast Cytomegalovirus cerevisiae having hyper rapid growth rate, simple nutritional requirements required, can be grown on semi-solid agar. Mutant strains can be isolated by replica plating. These are the bacterial clones on the left side, and the right side of the yeast clones. That looks like. And replica plating, for replica plating you have to just have sterile velvet covered stem, and these are the colonies, and then your missing colonies requires arginine minimal medium and minimal medium on that, so which lacks arginine and plus arginine in that case. So growth of animal cells in culture requires rich media including essential amino acids, vitamins, salts, glucose, and serum, and most grow only on the special soil surfaces. So they are primary cell line that we have discussed in detail about that already. So there are some fibroblasts can be cultured and differentiated, and then there's undifferentiated cells and differentiated cells that looks like this. Then there is a generation of transgenic mice and knockout mouse, loss of functions, gain of functions, and flavors. In the loss of functions, mainly are the knockout mice and siRNAs. In the gain of functions, there is a transgenic mouse, and in the flavors, there is conditional inducible systems and tissue specific systems. So there's organisms, which is genotype means its entire set of genes that denote whether the individual carries the mutations in one or more genes, and phenotype is its functions, physical appearance, and depend upon the individual genotype. So their normal organisms produce develop changes term mutations in their DNA sequence, thereby altering their genotype, perhaps their phenotypes. So there are haploids and diploids. Haploids having a single copy of each chromosomes, and phenotype sequence of that one copy. And diploid organisms have two copies of each chromosomes, and thus two copies of each gene. So diploids that carry uh, identical alleles are termed homozygous, and diploids that carry different alleles are termed as heterozygous. So alleles may be dominant or recessive. So diploid genotype includes, uh, so this is a wild type, dominant, and then recessive and recessive. So wild type looks like this one. If the dominant is present singly, then it will be mutant. And if they are present twice, it will be mutant. But if recessives are present, if single time, then it will be wild type. But if they are present twice, then they are mutants and also. And there are some genotypic linked diseases. So something like sickle cell anemia, cystic fibrosis, phenylketonuria, Tay-Sachs disease. These are all autosomal recessive diseases. And autosomal dominant diseases are Huntington disease, hypercholesterolemia, and there are X-linked recessive diseases like Duchenne muscular dystrophy, hemophilia, and their molecular and cellular defects are respectively. And this is segregation of genotype linked diseases, how segregately your genotype transfer to the next part. So here you can see autosomal dominant Huntington disease, where it has been your male part is being affected and female part is not affected. So they are male and females, right? So HD. So you can see the in the yellow color the affected ones, and the ones are the non-affected ones. So autosomal recessive cystic fibrosis are the one with the CFTRs with the carrier, and then these are the females are the carriers, and the males are also carriers here. And male and female, when both are present, then they are affected. Otherwise, they are carriers in the further generation. And in the X-linked recessive Duchenne muscular dystrophy, here you have a male as a carrier as a DMD disorder, and then you have again male as a disorder, and the rest are carriers or uninfected in that case. So it mainly are being transferred if it is present in the male or female. They are X-linked diseases. So there are mutants in Drosophila with the red eye and white eye, or some UBX mutant with the two wings extra being present here. And there is a gene replacement and transgenic animals. It is possible to replace an organism wild type gene with inactive gene to create a gene knockout. It is possible to produce additional genes to create a transgenic organism. So there is gene knockout, transgenic animal, and gene knockout and transgenic techniques involve mutagenesis of cloned genes prior to the transfer to the organism. So we need to mutate the organism before we could go ahead with that. So students, these are some creations of mice. First, you have to create mutation in the embryonic stem cells to carry out the knockout mutations. So here you can see specific targeting of gene of interest, formation of embryonic cell line carrying a knockout mutations. So you have gene X replacement construct, new R and TXHSV. Then homologous recombinations are present there, and then afterwards ES cell DNA is crossed with the gene targeted insertions, and mutation is done by resistance to G14 and Gannico clover. On the same right hand side, non-homologous recombination is done with addition of ES cell line DNA, and random insertion is done to make no mutation in the gene X resistance to G14 but sensitive to granocollular. Then further you have to do is positive and negative selection of recombinant ES cells with random insertions, non-recombinant cells recombinant with gene targeted insertions, and then you have to treat with G14 positive selections, treat with the Gannico, and you have targeted disruption in the gene X cells. Then you use those embryonic stem cells into the blastocyst of early embryos. So you have brown mouse with AA X minus X plus, yeah, and you have black mouse with small a recessive and X plus X plus. So you put them into black mouse with the brown mouse embryonic stem cells, and then you wait. You have a 4 to 5 day of blastocyst. Then surgically transfer the embryos into the pseudo pregnant female. So you take one female and you foster the mother. The possibly progeny we will have will be chimeric and black. And then we take the chimeric for the cross to the wild type black mice. So we cross with the wild type black mice with the chimeric. Possible germ cells will be AX plus, AX minus, and small ax plus. And all the germ cells of this will be black mouses AX plus. So ES derived progeny will be brown. So you will have brown progeny here and one black. Then from these brown progeny, from ES derived germ cells, we will screen for brown progeny DNA to identify X minus, X plus heterozygotes. Then you made X minus, X plus, screen progeny DNA to identify X minus, X minus. Then you knock out your mouse afterwards. So this is LOXP mouse, Cree mouse. All cells carry endogenous genes, sides flanking exons too, and then Cree mouse. So to know about them, let me so this is one more method about in vitro production of double-stranded RNA. So you have a sense transcript, antisense transcript. Then you have a gene of interest, GOI, and then you have a dicer gene to produce siRNAs to knock down your specific type of mRNA. So this you can do it to stop one gene from your further reactions. And you can insert them into the, this is non-injected and injected seems like. Then production of transgenic mice, then inject foreign DNA into the pronuclei of a fertilized mouse egg to fuse them for male and female pronuclei. Then transfer ejected eggs into the foster mother, and about 10 to 30% of offspring containing injected foreign DNA. Foreign DNA is present in equal amounts in all tissues, and mice expressing foreign DNAs are bred to continue in the germ lines. So example is transgenic mouse here. So this is the video about how things are done.
Gene knockout is a common technique in studying gene function. In this practice, the gene of interest is disrupted by altering the cell's genomic DNA. Any phenotype that results from gene knockout gives insight into the function of the disabled gene. Conventional knockout experiments are frequently achieved in animals, but need a huge investment of time, money, and labor. Therefore, these experiments are usually restricted to well-funded labs or core facilities. This has presented a large hurdle for researchers exploring gene function. The breakthrough of CRISPR technology has turned the tide and made gene knockout a much more accessible tool for many more research labs. The key revolution brought about by CRISPR is the creation of double-stranded breaks sequenced specifically. With CRISPR, scientists can create double-stranded breaks at virtually any site in the genome. To repair the double-strand breaks generated by CRISPR, there are two main mechanisms: non-homologous end joining and homology-directed repair, also known as homologous recombination. With non-homologous end joining, the ends of the double-strand breaks are simply reconnected together, and this process is highly error-prone, introducing mutations, insertions, and deletions, and in many cases resulting in gene knockout. Homology-directed repair, on the other hand, utilizes a repair template, where desired changes are flanked by left and right homologous sequences. Through homologous recombination, the desired changes are incorporated into the genome. The CRISPR knockout knock-in kits utilizes the homology-directed repair system. To plan an HDR-backed knockout experiment using plasmids, the researcher needs to create the following constructs. 1. A plasmid coding for the gRNA, targeting the intended site for double-stranded cleavage. 2. A plasmid coding for the Cas9 protein. And 3. A plasmid serving as the repair template, with a designed sequence flanked by the homologous arm region of the cleavage site, or donor vector. Origin has created pre-designed kits to make knockout experiments even easier. For each gene locus, we have created a specific kit. The first two elements are provided in a single all-in-one vector. The gRNA vector, targeting the start of the coding sequence, was cloned into a vector containing the expression cassette for Cas9 protein. The donor vector was also created with a knock-in GFP puromycin cassette, flanked by the homologous arms of the chosen cleavage site. This permits the selection of knockout cells via puromycin marker. The promoterless GFP provides additional utility to monitor any upstream promoter activity that might be present. To start your knockout experiment, simply co-transfect a gRNA vector with a donor DNA plasmid into the cells. The Cas9 enzyme will cut a double-stranded break near the start codon, and through homologous recombination, the functional cassette will be integrated into the genome, replacing target gene. In the end, 3 events are achieved. 1. The target gene is knocked out through gene disruption. 2. GFP will be driven by the endogenous gene promoter, providing added information of promoter strength. And 3. The puromycin marker under the PGK promoter is integrated into the genome, enabling the selection of successfully edited cells. The protocol starts with co-transfection. There will be 3 separate experiments, one of the 2 gRNA vectors 30 days before puromycin selection, as the donor vector alone before genomic integration also provides puromycin resistance. Culturing the transfected cells for 20 days ensures that the episomal form of donor vector is lost. This diagram shows the details of transfected cell passaging prior to puromycin application and selection. For HEK 392 cells, 48 hours post-transfection is considered G1 cells, at which point the cells are split 1 to 10 and grown for 3 more days. At G2, CRISPR-targeted cleavage and gene editing should be finished, but the percentage of edited cells will be small. You can perform optional genomic DNA analysis using G2 cells. If the data are negative, you can redo the analysis after puromycin selection. After G7, apply puromycin selection using the doses appropriate for your cells. To summarize the CRISPR knockout knock-in protocol described so far, Step 1. Co-transfect one of the gRNA vectors with the donor vector, along with the appropriate controls. Step 2. Passage transfected cells for around 20 days. Step 3. Apply puromycin selection and isolate individual cell colonies. Please note that the drug selection doses need to be determined by kill curve for each cell line. 5 days after puromycin selection of transfected HEK 293 cells, almost all cells in the 2 control transfections died. However, as expected, The co-transfection with the gRNA construct produced puromycin-resistant cells. The fourth step is to analyze the puromycin-positive cells to detect the knockout. If you have a good protein-specific antibody, you can use Western blotting to evaluate the knockout. It is best to use this method after isolating single colonies. Another method is genomic PCR to verify the correct GFP puromycin integration in the genome, followed by sequencing to confirm the integration. To amplify only the edited allele, design PCR primers following these rules. The forward primer should be upstream of the 5' end of the left homologous arm of the donor vector, and the reverse primer should be in the GFP region. In this example, knockout ATG5 gene was conducted by the protocol. Genomic PCR products of the correct size were amplified from the gRNA constructs, plus the donor co-transfected cells, and not from the scramble control transfected cells. The PCR products were further sequenced using the PCR primers to confirm the correct integration. Sequencing data using the forward primer shows the correct integration of the 5' end of the left homologous arm in the donor vector. The reverse sequencing primer confirmed the correct integration of GFP puromycin cassette. The original ATG5 gene was replaced with GFP. Origene offers genome-wide human and mouse CRISPR knockout knock-in kits. With these complete, ready-to-use kits can knock out almost any gene from the genome and knock in the GFP and puromycin cassette simultaneously. You can study the consequences of the gene disruption and potentially do a promoter study at the same time. Origene also has other functional cassettes to provide different fluorescent proteins, luciferase, or different selection markers. If you wish to have the CRISPR knockout kit created a different donor vector, please visit our website to request a custom project. To learn more, please visit the Origene website and our YouTube channel.
So that was about a short description about CRISPR-Cas9 also. That is very important nowadays. People are working on that thing. And so I hope. What is this stupid things? How to report these people? So that was about CRISPR-Cas9.
The ability to insert or knock in a gene at a specific locus is a tremendously useful method for dissecting disease mechanisms. However, in previous years, the insertion of a foreign gene has been far too tedious and expensive for many labs to perform. Thanks to the recent development of CRISPR-Cas9 technology, gene knock-in experiments have now become a very popular technique in modern molecular biology. In contrast to the random integration events by the lentiviral system, CRISPR utilizes a guide RNA that directs gene insertion with extraordinary specificity. In particular, the AAV-S1 safe harbor site is often a preferred target site for gene knock-ins. This is because it enables consistent and robust transgene expression with minimal toxic effects. At this safe harbor site, the surrounding DNA maintains an open conformation, enabling stable expression of the newly inserted transgene. This makes safe harbor sites, such as the AAV-S1 locus on human chromosome 19 and the ROSA 26 locus on mouse chromosome 6, very popular sites for targeted knock-in experiments. In this case study, we will walk you through how a red fluorescent protein gene was knocked into human embryonic kidney cells at the AAV-S1 safe harbor site. We will also demonstrate how the knock-in was verified using a PCR-based method. To begin, an sgRNA was designed to target the human AAV-S1 locus. Software analysis was performed to ensure maximum efficacy of on-target cleavage while avoiding any off-target effects. The resulting guide sequence was used to construct ABM's AAV-S1 targeting all in one vector. This vector expresses the Cas9 endonuclease under a CMV promoter and the sgRNA under a U6 promoter. To achieve the knock-in, a donor plasmid containing the gene of interest is required to provide a template for homology-directed repair. The HDR pathway is a mechanism in cells that repairs double-stranded breaks by using a DNA repair template homologous to the sites flanking upstream and downstream of the break site. To enable HDR-mediated gene insertion, the gene of interest must be flanked by such homology arms. Therefore, a second plasmid was constructed by cloning an RFP pyromycin expression cassette into ABM's repair template plasmid for the AAV S1 locus. The resulting AAV S1 donor RFP plasmid had the RFP gene under a CMV promoter and a pyromycin's resistance marker under a PGK promoter. Once both plasmids were ready, they were introduced into HEC293 cells using ABM's transfection reagent, DNA fectin. Following transfection, the cells were passaged 10 times to dilute out the episomal donor vector. Following, the resulting HEC293 cells were given 3 to 4 weeks to grow in the presence of pyromycin to select for the cells with successful knock-in of the RFP pyromycin resistance cassette. Cells transfected with both AAV-S1 targeting all-in-one vector and the AAV-S1 donor RFP plasmid were healthy after 4 weeks, and greater than 95% of HEC293 cells were successfully expressing RFP. Control cells that were not transfected with the vectors died after pyromycin treatment. PCR genotyping was carried out to confirm that RFP was successfully knocked in at the AAV-S1 site. In this method, 2 primers were designed. Primer 1, targeting the 5' homology arm upstream of RFP, and Primer 2, targeting a site within the RFP pyromycin resistance cassette. Gene knock-in was confirmed when a PCR product of 1.1 kb was observed, indicating that primers 1 and 2 were both able to bind to the edited genome to enable successful PCR amplification. In the case of a non-successful knock-in or in the presence of wild-type control cells, no PCR products were observed, as primer 2 could not anneal to the target site and enable amplification. We hope this case study has demonstrated how the CRISPR-based gene knock-in technique can make your exploration of gene function amazingly easy and efficient. CRISPR-Cas9 mediated gene knock-in allows for biallelic modifications of some genes to be generated with ease, with phenotypic changes in mice observable in a single generation, an unprecedented time frame. Let ABM's team of CRISPR scientists help you knock in any gene into any cell line you want, all with minimal toxic effects at the AAV S1 or ROSA26 safe harbor sites. Explore ABM's extensive portfolio of customizable CRISPR services and products today, and start knocking in genes, tags, markers, and more. To make it even easier for you, simply describe your desired knock-in, and we'll take care of the rest.
So that was about CRISPR. Cas9 in quite details. I felt that has to be teach you in detailed manner. So we are done with this lecture today. Finally, and so Bachcha, your question is during pyrosequencing, how we came to know that signaling obtained is due to the luciferase activity or due to the ATP molecules that are failed to take part in the strand synthesis. So I can't answer that actually. If anybody knows, please answer to that question. I can answer it, I can write it in my notebook and will answer to you tomorrow if possible. So I will write in my notes. Okay. So I will look for the answers for that, students. Don't worry. So, shall we have a break now for 5 minutes? or 5 to, yeah, we meet at 20 at 9:05. We meet at 21:05 then, so that you can take some break, and we start again with the today's lectures of next topic, that is RFLPs, AFLPs, RAPDs, SSRs. So they are quite important for us, so we try to start with them today. So let me just take a quick break and then we start at 9:05 then. So DNA based markers, so RFLP, AFLP, RAPDs, SSRs and MAS. So we will go through each of them in details. So, so appendix looks like we will go through molecular markers, basic principles of molecular markers detections, molecular marker based on DNA hybridizations, markers based on PCR amplifications, random amplified polymorphic DNA, amplified fragment length polymorphisms, AFLPs, and molecular marker assisted selections, mass selection. So we will go through all of them today. So molecular marker is a DNA sequence in the genome which can be located and identified. So as a result, genetic alterations like mutations, insertions, deletions could be located in genome by different plants. And these differences are called polymorphisms and can be mapped and identified. And we can detect them and we call them as molecular markers. So molecular markers are highly reliable and advantageous in the plant breeding programs because they represent genetic makeup at the DNA level. They are consistent and not affected by environmental factors. Thirdly, they are detected much before the development of plants occurs. So large number of markers are generated as per the needs. And the basic principle of molecular marker detection, what you have to do is, let's assume there are two plants, one is disease sensitive and other is with disease resistance. If there is a DNA marker that can identify these two alleles, then genome can be extracted, digested by restriction enzymes and separated by gel electrophoresis. So DNA fragments can be detected by their separations. For instance, disease-resistant plants may have a shorter DNA fragments, while the disease-sensitive plants have longer DNA fragments. And molecular markers could be divided into two types. One could be DNA hybridization based on the DNA hybridization, that is non-PCR based, and other is based on the PCR amplifications, PCR based. So this is the molecular markers identification looks like. These are disease-sensitive plants and these are disease-resistant plants. So DNA extraction is done on this side, DNA extraction on this side. So then digestion of friction enzymes of gel electrophoresis, then there is a shorter length from the disease sensitive plants and there is a longer DNA fragments from the disease resistance plants. So this is how after the friction enzymes gel electrophoresis, you have shorter and longer DNA fragments. Then molecular markers based on DNA hybridization. So DNA pieces can be cloned and allowed to be hybridized with the genomic DNA, which can be detected. So markers based DNA hybridization is widely used. The major limitation of this approach is that it requires large quantities of DNA and use of radioactivity. So restriction fragment length polymorphism, RFLP, are the very first technology employed for the detection of the polymorphism based on the DNA sequence differences. RFLP is mainly based on the altered restriction enzyme sites as a result of mutations and recombinations of genomic DNA. So, an outline of RFLP analysis is given in the figure 53.2 and 53.3. So, in this, we mainly do the isolation of genomic DNA and then digestion of by restriction enzymes, then separation by electrophoresis, then finally hybridization by the incubating with the clones and label probes. So you have your isolation of genomic DNA and then you digest them with the restriction endonucleases. Then you have DNA pieces, then they separate them by agar gel electrophoresis and DNA pieces transfer to the membrane filter. Then they are incubated with the clone and label probes. Then you have RFLP bands detected there. So based on the presence of friction sites, DNA fragments of different lengths can be generated by using different restriction enzymes. So in the figure 53.3, so you can see two DNA molecules of the plant A and B are shown. So in a plant A, mutations occur leading to the loss of friction sites that can be digested by E. coli. So there is a mutation happen, sorry for my voice, there is a mutation happening in the plant A, so that the E. coli is not restricting it, but in the plant B it is restricting it. So after restriction of Hint3 enzyme, there is a 200 base pair and 800 base pairs of two DNA fragment segments. But in the hint 3 you have 200, 450, and E-coli you have 350 and 650. 350 and 650. So you can digest them with restriction enzymes, and then with the enzyme with hint 3 that you see is that 800 and 200 base pairs monomorphic pattern. But in the case of with the E. coli, you see in the case of plant B you see two different patterns, and in pattern A it's not working, so it's 1000 base pairs. But in this, so this is a polymorphic pattern. So that's why these results are called as polymorphic patterns of separations. That is friction fragment length polymorphism. This is also showing you plant source, DNA, friction digestions, electrophoretic separations, southern hybridization, then autoradiography is done afterwards. Then marker based on PCR amplifications. I need a cup of tea or something that my voice is fine. Um, so polymerase chain reactions based, we are done with the DNA hybridase based. Now we are going with the PCR based polymerase chain reactions, which is a novel technique for the amplifications of selected regions of DNA. So the advantage with PCR is that even a minute quantity of DNA can be amplified. So thus PCR based molecular markers requires only a small quantity of DNA to start with. So the result is that the PCR based markers may be divided into two types, that is local non-specific and local specific markers. So local non-specific markers are example are random amplified polymorphic DNA RAPDs and amplified fragment length polymorphism AFLPs. So local specific markers, that is simple sequence repeats, single nucleotide polymorphism SNPs and SSRs. So let's start with RAPD first, random amplified polymorphic DNA markers. So in them, what you do is RAPD is a molecular marker based on the PCR amplifications. So an outline is given here. So you have to isolate the genomic DNA. Then you denature that DNA, then anneal with the template with the primers, then complementary standard synthesis is done, then amplified products are being seen by the gel electrophoresis and identified later on. So single short oligonucleotide primers can be arbitrarily selected and used for amplifications of the genome. Then amplified products are separated on electrophoresis and identified. So applications of RAPDs in plants. So RAPD is used to distinguish between variety, is based on the difference in the DNA sequence, and RAPDs have been used to identify nearly 15 commercial sunflower varieties. The new bean varieties, which are difficult to distinguish between the morphological traits, have been ideal for the candidate of the applications of the RAPD markers and methods. So this produced 296 markers, and that could be scored. Almost 85% similarity was predicted. For example, a DNA plant allows the amplifications of sequences of A, C, D, but not B. So A, C, D is being amplified, but B is not being amplified. So we see only these two paths. So this is how RAPD markers works out. So RAPD markers have been used to identify several disease resistance genes in plants. RP94 gene is responsible for resistance to stem rust in barley, and RAPD markers identified to link to these genes. Similarly, RAPD markers linked to the heat smut resistance genes have been characterized. So controlling of height in barley plant by specific genes have been used to locate dwarfism gene by RAPD markers. Are not required. So there are some demerits of RAPDs. They are constrained about reproducibility of results. Since RAPD markers are dominant, only half of the genetic informations are codominant markers. Null alleles are not directly directed in them. So based on the nucleotide alteration in the genome, the polymorphism of amplified DNA sequence differ, which can be identified as bends on the gel or electrophoresis. Genomic DNA from two different plants often results in different amplifications patterns, that is RAPDs. This is based on the fact that particular fragment of DNA may be generated from one individuals and not from the others. So this represents polymorphism and can be used as molecular markers of a particular species. So AFLP is a novel technique involving a combination of RFLP and RAPDs. AFLP is based on the principle of generation of DNA fragments using restriction enzymes and oligonucleotide adapters and their amplifications by PCRs. Thus, this technique combines the usefulness of restriction digestion and PCR both actually. So here is an example done here. So you have ACTTA, GAAC, and you have CATAATTT. Then you have a DNA molecule here. So you restrict them, these enzymes, with E. coli 1 and MSC1. So you have left side and right side. And then you CTTA, GAC, then CATA. These are removed. So from the left side 6 are cutted out and from the right side 4 are cutted out. Then you ligate with the linkers are added, then pre-selective amplifications of employing single nucleotide extension, then in further the opposite sequencing will be started to increase, and selective amplifications of employing the additional nucleotide extension. So they are extended like this further. The same thing is shown on the right. You have isolated your plant DNA, you first restrict them, then you ligate them with adapters, then add non-specific primers and perform PCR. Then you have electrophoresis, then you can visualize them like this. So AFLP is primarily used in the genetic mapping. Several economic important factors are there such as rice, barley and wheat and have been mapped by AFLPs. So AFLP markers exhibit strong relation between number of markers per chromosome and length of the chromosome. Similarly, in rice, cross between Indica japonica reveal that 50 AFLP markers are there located on every chromosome except in the small chromosome 12. And also they are comprises of around 157 RFLP loci, which has been increased by adding 118 AFLP markers to that. So there are some merits. AFLP is used to screen large number of polymorphism. It is possible to saturate specific regions of genome for map-based cloning of target genes. So AFLP has also been used in phylogenetic studies and distinguishing features between varieties. So AFLP is also used to screen pools of plasmid DNA from several clones, and they have been applied to analysis of quantitative traits in barley and rice. So image analysis of fluorescent PCR products developed by key genes, and they have many research of it. There are some demerits also, not reliable to convert AFLP into scars. We will discuss about scars soon. So null allele cannot be detected. Proprietary techniques are require high amount of DNA that required in RAPDs, and silver staining and radio labeling is also very expensive to do so. Now comes sequence tag sites. So they are the one which are uniquely simple copy segments of genomes whose DNA sequences are known and can be amplified by using PCRs. So STS markers are based on the polymorphism of simple nucleotide repeats and have been recently developed in plants. So when the STS loci contains simple sequence length polymorphism SSLPs, they are highly valuable as molecular markers. So STS loci have been analyzed and studied in a number of plant species. So microsatellites are the randomly repeated multi-copies of mono, di, tri, tetra nucleotide motifs. In some instances, the flanking sequence of the repeat sequences may be unique. So primers can be designed for each flanking sequence to detect the sequence tag microsatellites, and this can be done by PCR. So sequence characterized amplified regions are modified forms of STS markers, and they are developed by the PCR markers that are made of the RAPD fragments. So they are sequence characterized amplified regions. They are the modified forms of STS markers, and they are developed by the PCR's primer that are made up end of RAPD fragments. So STS converted RAPD markers are sometimes referred to as scars also. Then comes the molecular marker assisted selections. So these are the ones, the selection of desired traits and improvement of plants of crops has been part of the conventional breeding programs. So this is predominantly based on the identification of phenotypes. It is now accepted that phenotypes do not necessarily represent the genotypes, so many times the environmental make the genotype at the end. The following major requirements are there for the molecular marker made in plant breeding. They should be closely linked with desired trait. They should be efficient, reproducible, and easy to carry out, and analysis should be economical. Further, their molecular breeding helps to yield and quality, and they represent the breeding methods that are coupled with the genetic engineering techniques. So they help in the grain yield and cereal productions. So their development of dwarf and semi-dwarf varieties of rice and wheat have been responsible for the green revolution. La la la. So yeah, this one is important. Quantitative trait loci (QTLs). So there are many characteristics controlled by several genes in complex manners. So some good examples are growth habits, yield, adaptability to environment, and disease resistance. So the location on the chromosomes for these genes are regarded as a quantitative trait loci (QTLs). And major problem with them the plant breeder faces to how to improve the complex characteristics to manipulate the multiple genes in the genetic engineering. For instance, development of a golden rice involving the insertion of just 3 genes took about 7 years. So thank you very much students. That was about RFLP, AFLP, RAPDs, SSTs, molecular marker assisted screening of your uh, data. Now I will teach you a little bit about restriction enzymes. So before that, all is fine with my students. Okay, let me just grab warm water so that I could be fine with the lecture. Just give me 1 second. I'm coming back. Okay, let's start with restriction enzymes. So mainly we will deal here with the nomenclature of restriction enzymes. What are the various types? What are the various functions? And then so that's the appendix of it. We will go through the introduction, mode of action, star activity, nomenclature of restriction enzymes, type of restriction enzymes, DNA ligase, kinase activity, reverse transcriptase, and clinofragments. So the ability to uh, manipulate DNA into in vitro depends entirely on the availability of purified enzymes that can cleave, modify, and join the DNA molecules in the specific ways. So at present, no chemical methods can achieve the ability to manipulate the DNA in vitro predictable way. So only enzymes are able to carry out the functions of manipulating the DNA. So each enzyme has a vital role to play in the process of genetic engineering. The various enzymes used in genetic engineering are as follows: nucleases, restriction enzymes, DNA ligase, kinase, phosphatase, reverse transcriptase, terminal deoxynucleotide transferase, and RNAPs. So nucleases are the group of enzymes which cleave or cut the genetic material, that is DNA or RNA. So DNAs and RNAs are the further classified into two types. Nucleases are further classified into two types upon the substrate they act. They could be acting upon the DNA, which cut on or cut the DNA as classified DNases, which cut on the RNAs, they are called as RNases. So our DNAs are further classified two types according to the position they cut. They could be at the end they are cutting, or terminal region of DNA, or they are called as exonucleases. The one which are cutting at the center, which are not specific region, they are called endonucleases. So exonucleases require a DNA strand with at least two 5 end and 3 prime end, and they cannot act on DNA which is a circular. And endonucleases can act on circular DNA and do not require uh, any free can act on the DNA or free DNA exonucleotides. So restriction enzymes which act on the specific position or sequence of DNA are called restriction endonucleases, which act on specific sites, so they are called as restriction endonucleases. And the sequence which are recognized by the restriction endonucleases, or we also call them restriction enzymes, they are called as recognition sequences or restriction sites. So these sequences are palindromic sequences, like they have the same meaning from the left to right and right to left, like ATA, ATTAA, then AATTDA. So both sides are same. So different restriction enzymes present in the different bacteria can recognize different sites, but they will cut two different within the restriction enzymes, and they are called isomers. And instantly, two restriction enzymes from a single bacteria will cut at the same restriction sites always. So how they work, how their functioning is done. So they are mainly bind to the recognition sites, check for the methylation first, and if the methylation is recognized in the recognition sequence, then it is just falls off the DNA and does not cut. If only one strand in the DNA molecule is methylated in the recognition sequence, the other strand is not methylated. So then restriction enzymes will methylate and other strand at the required positions. So the methyl group is taken by the RE from the S-adenosylmethionine by using modification sites present in the restriction enzymes. However, type 2 restriction enzymes takes place with the help of another enzymes called methylase and methylate the DNA. Then restriction clears the DNA. If there is no methylation on both strands of DNA, then restriction enzymes cleaves the DNA. It is only by this methylation mechanisms that restriction enzymes, although present in bacteria, does not cleave the bacterial DNA but cleaves the foreign DNA. So like this they work, and there is a star activity called as sometimes enzymes recognize and cleave the DNA strand at the recognition sites with asymmetric palindromic sequences. For example, BAM H1 cuts the sequence at GATCC, but under extreme conditions such as low ionic strength, it will cleave at any of the following sequences, NGA, TCC, any of these. So such an activity of the RE is called star activity. So this is the nomenclature of friction enzymes. So as a large number of friction enzymes have been discovered, a uniform nomenclature system is adopted to avoid confusion. So this nomenclature was first proposed by Smith and Nathans in 1973, and every friction enzymes would have specific name which would identify it uniquely. The first three letters in italics indicate the biological source of the enzymes, and first letter being the initials of the genus and the second and third being the first two letters of the specific species name. So thus restriction enzymes names are given from Escherichia coli called ECO first, then Haemophilus influenza becomes HIN, and Diplococcus pneumonia becomes DPN. Then comes the letter of the strain of the bacteria, that is ECO R, strain is R. Then comes the Roman numeral of that strain sequence with the E. coli 1 for the first enzyme, for the second it's E. coli 2. So that's how you give the naming to the restriction enzymes. And there are three types of restriction enzymes. They are divided into type 1, type 2, type 3. Type 1 and 3 have an ATP independent restriction activity and modification activity which are resident in the some multimeric proteins. Type 1 enzymes cleave the DNA at random site, whereas type 3 cleave at a specific sites. Type 2 restriction modification systems possess separate enzymes for endonucleases and methylase activity, and are the most widely used for genetic manipulations. So type 1 restriction enzymes, these restriction enzymes recognize the recognition site but cleave the DNA somewhere between 400 base pairs to 10,000 base pairs. So these enzymes are made up of 3 peptides with multiple functions. These enzymes requires Mg++ ATP as adenosyl methionine for cleavage or for the enzymatic hydrolysis of DNA. So these enzymes are studied for general interest rather than the useful tools for the genetic engineering. So they are the various types. They are explaining about it, about various enzymes, and this is the common summary of that. Type 1, type 2, type 3, with enzyme complex of 500 to 600 kilodalton composed of 3 separate units. This is normally homodimers and these are heterodimers. And this is multi-enzyme complex and subunits. This is separate enzymes, endonucleases, homodimer, methylase, and monomer. And subunit provides specificity of its own function as methylase and a heterodimer with R subunits. And cofactors are magnesium, ATP, acetyl, SAM. Here is also magnesium and SAM. Here is magnesium, ATP, and SAM, all three. So that's how the various types of REs are. Then comes the various types of how they're cutting. Based on the cutting, there are 4 base cutters, 6 base cutters, and also 8 base cutters. So 4 base cutters are GATC, CCG. So they are cutting the four base pairs. And there are also then 6 base cutters. Then there's 8 base cutters like this. NOT 1, SBF 1, PVU 1, PVU 22, CLA 1, BGL 1. Then DNA ligase, it's a recombinant DNA experiments require the joining of 2 different RNA segments or fragments in vitro. The cohesive ends generated by some RE will anneal themselves by forming hydrogen bonds. To get a stable joining, a DNA should be joining by using an enzyme called ligase. And DNA ligase join the DNA molecules covalently by catalyzing the formation of phosphodiester bonds between adjacent nucleotides. So then T4 ligase requires ATP as a cofactor, E. coli ligase requires NADP as a cofactor, and cofactor is first split and then AMP binds to the enzyme to form enzyme AMP complexes. Then there are kinases. Kinases is a group of enzymes which adds a pre-pyrophosphate to the wide variety of substrates like protein, DNA, and RNA. And it uses ATP as a cofactor and adds phosphate by breaking the ATP into ADP and pyrophosphate. So there are 2 types of, they are mainly alkaline phosphatases, and they are the group of enzymes which remove phosphate from variety of substrates. And phosphatases which act in the basic buffer with pH 8 or 9 are called alkaline phosphatases, and they are commonly known as BAP, bacterial alkaline phosphatases, and CAF, intestine alkaline phosphatases, BAP and CAP. And alkaline phosphatases is a metalloenzymes and has zinc ions in them. And bacterial alkaline phosphatases is a dimer containing 6 zinc ions, 2 of which are essential for the enzymatic activity. VAP is very stable and is not inactivated by heat and detergents. Whereas the CAF, intestine alkaline phosphatases, is also dimer. It requires zinc ions and magnesium ions for actions. Then comes the reverse transcriptase. These enzymes uses an RNA molecule as a template and synthesize a DNA strand complementary to the RNA molecules. And these enzymes are used to synthesize the DNA from RNA. And they are present in the most of the RNA tumor viruses and retroviruses. And they are also called as RNA-dependent DNA polymerase. Then comes the terminal deoxynucleotide transferase. These are the one which polymerase and adds which adds nucleotides at the 3' end, but does not require any complementary sequence and does not copy any DNA sequence. So terminal deoxynucleotide transferase as nucleotide whatever comes into its active site, and it does not show any preference for any nucleotides. So RNase piece specifically cleaves at 5' end of RNA, and it is a complex enzyme consisting of small proteins of 377 nucleotide RNA molecules. Then comes the last clinoid fragments, that is the E. coli DNA polymerase, consists of single polynucleotide polypeptide chains. The two fragments that are obtained from the DNA polymerase 1, is called clinofragments, and these fragments is a 602 amino acids in length, and function of clinofragment is to add nucleotides to the 3' end and 3' exonuclease activity. So clinoid fragments adds nucleotides by using complementary strands as preferences, and it cannot extend the DNA without the presence of the complementary strand. So if any nucleotide is added by mistake and base pair is wrong, then by using 3 to 5 exonuclease activity present in the clinofragments. Now comes the last part from today, DNA fingerprinting. Just a second. Just a second, students. My phone is being hanged actually. Okay, so I'm getting hundreds of. So I think I should switch off my phone. Lot of OTPs from different websites, Flipkart, KoiBB, which I'm not using now at the moment. So my phone has been hacked. So students, please take care. Um, so I'm confused now. There's some money in my Paytm, so might be they're trying to fetch that money and attach to it. Sorry. I will switch off my phone so that no transaction could happen. So DNA fingerprinting is done. Do you have any questions, students? Okay. So why use DNA fingerprinting? DNA fingerprinting is done to tell is a way of telling individuals same species are apart, we are different, and DNA sequences are variable and can therefore be used as identifying characteristics. So DNA fingerprinting has advantages over other source of evidences, and they are highly accurate and can be gathered from the trace crime scene evidence. So one way is that you restrict with the help of restriction fragment length polymorphism, and the molecules that can cut into DNA pieces. Each enzyme cut at very specific DNA sequence, as we have discussed just now. While all human beings share roughly the same DNA sequence, there are in fact small number of differences, and these differences can be seen by restriction enzymes. So this is the RFLP animation. So this is BAM H1 is the restriction enzymes. It come and it starts to cut it out. Leads to production of sticky ends. Then this is individual one where four restriction enzymes are coming and they are cutting it. Production of five restriction enzymes, five fragments. Then individual two, A, B, C, D, and they are cutting it, and then we have like this, individual one and individual two. So essentially, once the DNA has been cut by the enzymes, we will have different DNA fragments of various sizes. Each individual's binding patterns should be different because the restriction enzymes will cut each person's DNA at different points. And fragments of different sizes will travel different distances along the gel, and we can see them through the electrophoresis gel. So how this is banned, these are the molecular markers, and here where samples are running from person 1, person 2, person 3, each showing individually different fragment of DNA. So each are having different fragments of length, it's different in their cases. So now how you can use them in the fingerprinting and forensic analysis. So forensic science can be defined as an intersection between the law and science. And the first photography then fingerprinting was done in 1985, and that is known as DNA fingerprinting. So DNA fragment shows unique patterns from one person to the next and used to interpret paternity disputes and as a forensic evidence. So specimen collection could be liked by envelope, dirty laundry, and cigarette butt saliva. So special precautions has to be taken care while doing so. Gloves, disposable instruments, avoid talking, sneezing, avoid touching samples with your skins, air dry the evidence from the package. So enemies of evidences are sunlight, high temperature, bacteria, moisture. Ideal samples are 1 ml of fresh whole blood treated with EDTA. So restriction fragment length polymorphism is help, as we have discussed, to cut individual 1 and individual 2 from the specific sites, so that individual 1 had 3 fragments and individual 2 has 2 fragments. So this is how RFLP is done, and this is how, if somebody wants to check the DNA, southern blot is done for the same to check the gels on the agarose gel. The next step is to blot transfer single stranded DNA fragments onto a nylon membrane. We will do about southern blotting in detail soon. And then there is a variable tandem number repeats, sequences that are repeated multiple times, and number of repeats varies from person to persons. So there are 5 repeats and 2 repeats. So VNTRs usually occur in introns, and VNTRs can be amplified by PCRs, and they are run on agarose gels to produce unique DNA fragments, fingerprints. Then this is done to amplify the segment. If the DNA is less, then you do the PCR to amplify it. We have discussed about PCR in details already, how we have annealing, denaturing, and then elongation method. Um, so I will not discuss this. So you can diagnose many diseases with this. Paternity testing you can do, forensic science you can do. You can check between crime scene, victim, and suspect with the help of DNA fingerprinting animation. This is not required. So that's it for today. So I will share all these lectures with you. So yesterday I shared the previous lectures already to you. So today I will share the other ones of today's day 4 workshop. Students, do you want to continue further? We can do for half an hour one more lecture. You can take 5 minutes break. We can continue if you want, if we have energy. Otherwise, what I can thinking is that the 3 days workshop will be switched to future days. I have to get internet somewhere, and then I can extend it to some next 3 days. Or otherwise, we can start putting half an hour till 10:30. Officially is 10:30. We can work till 10:30 today and can finish some work from tomorrow also. So I will try to match up everything together. So those whosoever are present, please confirm me with yes or no. Whatever you feel comfortable, we go accordingly that. Shall we extend the class or today, or we take 3 days extra somewhere? And please subscribe to my channel. Thank you so much. So whosoever has subscribed so far, others, those who have not subscribed, please subscribe it. It will really appreciate my future work. Okay, then 3 days extra, 3 days extra. Yeah, it will be difficult for you. So okay, so from 1 onwards I will be not taking classes, so I will be out of station from here. Some says extra is fine, some says 3 days extra. So mostly there's majority of 3 days extra here. So okay, majority wins. So we have a democracy in India, so we work like that, and democracy wins. So please, it will be extended to some days then. I don't know for how many days, but probably not 3 to 4 days. Yeah, yeah, I understand. No, no, I'm not going to Germany. No, no, I am in India itself. So I have some personal work. Just going for the same. So I'm not sure if whether that place will have internet or not. So I'm just concerned before I leave. So I don't want to put you all students into some pain that vice star left us like that. So okay students, good night. Wish you all the best for tomorrow, and those who have exams are coming, all the best. My best wishes for you, and enjoy your evening. Good night. Take care. Yeah, yeah. So type 2, why we use type? Because this is the one which actually cuts the things in the restriction sites very well, and they are the mainly famous ones actually, and they are widely present in the market also, and they're cheaper also. That's the main reasons to use restriction type 2 enzymes. Yeah. Okay. If somebody's questions are left, please write me on WhatsApp. I will try to answer them also. So for the moment, I wish you all good night. Take care. Bis dann. Tschüss. Bye bye. Have a nice evening. Sleep also. Good night. Take care.